Deoxyribonucleic acid is the name for DNA. We have all known about DNA for a considerable length of time, yet what do you truly think about it? What is DNA made of? In this paper we will discuss this smaller than normal supernatural occurrence called DNA. Like any great story, we have to begin toward the starting. DNA was found in 1868 by a Swiss restorative understudy named Johann Friedrich Miesser. It is imperative to note that Watson and Crick were the ones to dig more profound into the DNA setup. What they would realize is that the structure is really a coiled double helix on a 34-degree pitch. Hollick, 1995. They would encourage find that DNA is a long complex polymer produced using rehashing nucleotide. The two-fold helix DNA structure is produced using substituting sugar and phosphate bonds. Holding these sugar and phosphate bonds together is hydrogen. There are 4 base materials that are found in all DNA. These are adenine, cytosine, guanine and thymine. Simon and Dicker, 2012. With these 4 bases joined to the sugar, phosphate, they shape the complete nucleotide that we know as a DNA atom. DNA is set up at conception and never shows signs of change for the duration of our lives. It can totally copy itself, which permits it to have the capacity to copy and jump itself an interminable number of times. Despite the fact that it replicating itself a large number of times, the DNA is dependably a careful imitation from the earliest starting point. It is these 46 chromosomes that make up the complete guide for all that you are. Since half of the chromosomes originate from every guardian, that is the thing that considers every one of the varieties in our posterity. Researchers have said the human genome is involved roughly 3 billion DNA atoms. By and large, we are comprised of around 30,000 unique qualities that are spread between those 46 chromosomes we just discussed. With this a wide range of qualities, there are a limitless number of varieties that 2 guardians can make up. Every one of these chromosomes that make up our DNA are wound up inside the core of a eukaryotic cell. Besides simply the conceptive cells, every single cell contains the 46 direct chromosomes we specified. Of those 46, there are 23 sets of chromosomes. Of those 23, 22 are comparable in size, shape and even hereditary substance. The 23rd chromosome is the exceptional one. It is known as the sex chromosome. It is comprised of either 2 X chromosomes or an Z chromosome mix, and this is the thing that decides the sex of a person. Females yield 220 chromosomes, while males have the Z mix. DNA replication is basic to our survival. Every last new cell must have a careful reproduction of the DNA that was available in the first cells. This is our inherited outline maybe. This diagram is essential basically in light of the fact that this is the aid or chief to cell action and general organic improvement. As indicated by deoxyribonucleic corrosive, DNA, 2012, DNA sequencing in people is remarkable. In any case, of the around 3 billion base matches that finish the human genome, there is short of what 1% that is really distinctive. Our nearest living species is the chimpanzee. They share around 96% of our DNA. Despite the fact that that may appear to be greatly close examination, chimpanzees still have more than 40 million diverse DNA atoms. DNA additionally controls this different procedure called translation. DNA encodes particular attributes of a life form. Every trademark or attribute is then encoded in an exceptional area of the DNA cell called the quality. The quality for a specific attribute experiences another procedure called interpretation. This is a procedure that interprets the cell's dialect to another cell's dialect, DNA to ribonucleic corrosive or RNA, protein synthesis, which the body will comprehend and use to do the following procedure. The following procedure is the interpretation of the data from RNA to the real item, which is the protein blend. Protein combination is refined in the cytoplasm. After DNA is translated into a detachment particle known as RNA, a procedure that happens amid interpretation. The RNA should on the other hand be meant to produce a protein. For this interpretation to happen, ribosomes must cooperate deciphering RNA into proteins. Proteins are the workhorses of the cell. In the event that protein generation were to be frustrated, the cell would die. If this somehow managed to happen crosswise sufficiently over cells, then whatever these cells are a piece of will begin to die. If our cells quit making proteins, then we would be helpless to sicknesses like these and some more. Proteins and compounds do their work by synthetic procedures in the cell. A compound is a protein atom that works by setting off a substance response in cells all through our bodies. Each catalyst has its own particular trademark shape and influences just a specific sort of material. Without these impacts from compounds, our body would not have the capacity to change its interior chemicals sufficiently quick for us to maintain life. A few researchers see the human body as a continuous arrangement of compound responses. Compounds acquire an exceptional spot in our bodies by accelerating these responses, some of the time by as much as 1,000,000 times the rate at which our bodies would ordinarily work. Without this unbelievable quickened rate of action, there basically would not be sufficient time or responses for us to continue living. Sometimes proteins are likewise fit for fixing a portion of the responses in our bodies to return substance levels back to typical. Compounds assume a basic part with regards to sustenance in our body. Nourishment is our body's capacity to metabolize and utilize sustenance. This is done using proteins, whether those chemicals be from the nourishments we eat or from our own cells. Our body's cells are customized to manage every supplement to join and interface with different supplements and chemicals to make yet more chemicals, and which will thusly be utilized to assemble as well as repair the body's cells, bones, organs and the different tissues. Each metabolic response is begun, controlled and ended with a utilization of compounds. In conclusion, we talked about that was in 1868 when Johann Friedrich Miesser found DNA. We talked about that DNA comprise of 46 chromosomes and the 30,000 qualities spread between those 46 chromosomes. At that point we discussed the significance of protein union and how we would bite the dust without this procedure. At long last we clarified about the three sorts of chemicals and how their parts are essential to managing life as we probably am aware it.